I am Sunita Manna. Welcome to my channel, Study with Sunita. In this video we will discuss about gram staining. Gram staining is a differential staining. Gram stain was originally devised by Christian Gram. Now we will discuss about the aim of gram staining. The aim is to identify and classify gram-positive and gram-negative bacteria. The reagents are crystal violet, iodine solution, 95% ethyl alcohol and safranin. Principle. The gram stain reaction is based on the difference in the chemical composition of bacterial cell walls. Gram-positive cells have a thick peptidoglycan layer, whereas the peptidoglycan layer in gram-negative cells is much thinner, surrounded by outer lipid-containing layers. The procedure is as follows. Observation of gram staining. Bacteria that are gram-positive appear purple to blue, and bacteria that are gram-negative appear pink to red. The different shapes of bacteria are given below.